Chapter 7, DNA Recombinant. Nur Syafrina,
Nur Atika, Nabila Fantinurina, Nurul Batrisha,
Nur Farah Atira. Hello there, we are from
131
and our madam is
Madam Anissa Abubakar.
Definition of recombinant DNA technology, a method in which DNA molecule made in vitro with sequence from different sources or by combining genes from different organisms.
There have 5 tools used in recombinant DNA technology. First is target DNA, restriction enzyme, DNA cloning vector, host cell, and modifying enzyme. First is target DNA. Target DNA means sequence of DNA containing gene of interest. Example, gene encode for protein which can dissolve blood clots used in heart attack therapy.
Second is restriction enzyme. Restriction enzyme cuts DNA molecule at restriction site to produce restriction fragments. It also has short base pair sequence. It also contains palindromic. Restriction enzymes produce 2 types of cuts. One is sticky end and the other one is blunt end. Next is DNA cloning vector. DNA cloning vector has a characteristic of able to accept foreign DNA in MCS, that is multiple cloning site. It also able to replicate freely in the host cell. It also possesses selectable genetic marker. For example, in our syllabus is plasmid EGPUC18, DNA molecule that can carry foreign DNA into host cell and replication.
Number 4 is host cell, a cell which receives a recombinant DNA and allows the replication of recombinant DNA. Its characteristics: able to receive recombinant DNA through transformation process, able to maintain the structure of recombinant DNA from one generation to another, able to amplify the gene product from the recombinant DNA. Bacteria is the most commonly used as a host cell. Reason, DNA can be easily isolated from or introduced into a bacterium. Bacterial cultures grow quickly, rapidly replicating any foreign genes they carry. Example is E. coli. Number 5 is modifying enzyme, DNA ligase. Join the foreign DNA fragments containing gene of interest and bacterial plasmid by catalyzing the formation of phosphodiester to form recombinant DNA. There are 5 steps in gene cloning. First, isolation, cleave, insertion, transformation, and the last one, screening. Isolation. Plasmid from bacteria cell is isolated. Gene of interest is isolated from the donor cell. Second, cleave. DNA donor and plasmid bacterial is cleaved using the compatible restriction enzyme. Make a single cut sticky end. Third, insertion. DNA fragment insert into plasmid, joined together by DNA ligase, forming recombinant DNA. Fourth, transformation. The recombinant DNA then insert into the bacteria through its transformation process. All the bacteria reproduce, the recombinant DNA is also replicated. They have 3 recombinant DNA. Then the last one, screening. To identify bacteria clone containing recombinant DNA, blue-white screening is done. Plate of bacteria on agar containing antibiotic, ampicillin and sugar agar. Incubate until clones grow. Done.
Thank you for watching.